Hello, everyone. This is going to be the chapter on viruses and bacteria, and this should honestly be the last chapter of AP Bio. So let's get right into it. So when you're talking about like bacteria and viral infections, there are a lot of different types of viruses that are going to affect a lot of different parts of your body. You can see just a kind of host of these just to begin. And what we're going to talk about more specifically is in this section, viruses. So viruses are any infectious agent that cause a variety of diseases. They can be like this bacteriophage or the influenza virus seen here. There are many different types of viruses. These are too small to be seen under a light microscope. These are much, much, much smaller than what we can see as a with the cells. I always describe the viruses in my room. If you look around, those viruses would be the size if the whole room was the size of a prokaryotic cell, and the whole cell or a whole school was the size of a eukaryotic cell. So they are very small, even compared to prokaryotic cells. They're not really considered living because they don't, consist of all the characteristics of a living organism. But some people would argue that they are living. It's kind of a debate just within science and biology. You can see that size, how small it is compared to a prokaryotic cell or a eukaryotic cell. And there are many, many different types of viruses. We're not going to get into all the kind of dynamics of viral like parts, but we're going to go over some of the kind of basics here. You can see the Ebola virus. I think this is HIV. Yeah, this is HIV right there. So viruses, there are three main types or parts of a virus. You can see size and shape, the type of nucleic acid, whether it's single, double, or RNA or DNA, and the presence or absence of an envelope. And we're going to get into what those parts are. So the capsid, this is a protein coating that protects that DNA or RNA. This is just like almost like a protection or like armor for the virus. The envelope is a plasma membrane. It's made up of the previous host's membrane, because it used that membrane to kind of mask itself like a Trojan horse. But the membrane also has these viral spike proteins that kind of help it get into cells and infect them. You can see a virus without a envelope and a virus with an envelope. So we call these technically like naked viruses and an enveloped virus. So they're a little bit different, but they just either have a kind of plasma membrane or they don't. Glycoproteins, we talked about these months ago. These are proteins that allow access into a cell. They're on the outside of a cell. These act like keys. They're going to try to unlock the door into a cell. You can see what those glycoproteins look like. This is an actual image of it, so you can see those glycoproteins. Let's see here. In rabies, it binds to acetylcholine receptors as if it were a neurotransmitter. Cowpox virus binds to the epithelial cells as if it were a hormone, and some of these first viruses acted like glucose binding sites. So basically, what these viruses do is they act like already things that already exist and try to enter the cell by these methods. Bacteriophage, this is a type of virus that only infects bacteria. So you can see what these look like. I remember these being in Jimmy Neutron. That might be a little bit older for you. But these are a type of kind of like a spider-like virus, but they only attack and infect bacteria cells. You've heard of Elf on the Shelf. Now get ready for Nicolas Cage on the bacteriophage. Love it. All right, so I'm actually going to stop right there for a little bit. All right, 20.2 is going to look at how viruses reproduce, and these viruses are what they're going to do is they're going to act like a Trojan horse. So they're going to trick the cell's receptors to allow them into the cell. The virus is going to release its genetic material. Remember, that's DNA or RNA. The cell is going to use that genetic material to make new viruses. The cell doesn't know that it shouldn't make new viruses. It just makes whatever DNA or RNA sometimes is around. That's why we had to regulate DNA and RNA in chapter 13. So what happens then is the cell is going to burst, releasing the viruses that have been made within the cell. So I'm not going to show you this video, but you can see what happens here is the virus, now this is a bacteriophage, inserts its genome, it makes more of it, and it bursts, allowing those new viruses to infect other cells. This is for a prokaryotic cell, but it basically works the same for a for a sorry, I'm going to leave that in there. For a eukaryotic cell as well. Again, more videos we're not going to watch. So let me go back. One thing to kind of differentiate here, difference between the lytic and the lysogenic cycle. In the lytic cycle, the virus injects bacteria with which quickly replicates the virus and destroys the cell. In a lysogenic cycle, after the DNA is inserted, it's actually going to fuse with the host DNA, so it's going to become dormant. So this is a little bit hard to kind of understand as just a slide, but let's take a look here. In the Let's see here. The lytic cycle, you can see the cell is going to burst. However, within the lysogenic cycle, you're going to see that the DNA is fused within the cellular DNA already. So what happens then is the cell will divide, making many more copies, and that DNA will kind of remain dormant. So this is a way that there's two different life cycles of the virus, but this way it doesn't kill the cell. In the lytic cycle, it does kill the cell. Ooh, one more thing to talk about with virus reproduction, retroviruses. This is a group of RNA viruses that actually can take RNA back to DNA. This kind of goes against the central dogma of molecular biology. This is done by an enzyme called reverse transcriptase, which takes RNA and makes DNA from it. So this host cell will be infused with this DNA since it went from RNA to DNA, and it's integrated back into the DNA. So in this case, this goes from RNA to DNA and then DNA to RNA into proteins. This is a little bit different than what you've learned in your chapter 12 notes. All right, we're going to end there. 20.3 is going to look at subviral particles. These are a little bit different than viruses. They are not viruses, but they kind of act like viruses a little bit. Viroids, these are just strands of circular RNA that are going to infect cells, and they direct cells to make more of these circular kind of DNA. However, the circular DNA is actually kind of double-stranded as well sometimes. Man, these things are tough. They can cleave other RNA sequences, and they mostly result in plant diseases. And if you remember the potato famine in Ireland, this was caused by one of these viroids. But basically, it kind of acts like a virus, but it just doesn't. It's not a virus. The prions, these are very scary. They basically are unfolded proteins that make other proteins unfold. And it wouldn't seem like it's a bad thing, but these prions or unfolded proteins usually affect mammals in their brains, and there's no cure for it. You get it, good luck. There's no cure for these prions. Mad cow disease was an example of this, and this killed thousands of people years ago. And Kuru disease is actually an example of this, where the people of Papua New Guinea used to, as a ceremonial tradition, some of the people eat their dead, and it was a prion disease that kind of deteriorated the brain. Not kind of, it did, and you had the laughing disease because of one of the side effects was the brain deteriorated so much that the people would just laugh. So this ritual has since been stopped, and the disease obviously has then been stopped as well. But you have to ingest, or you have to actually eat prions to get them. This is not like you pass on through saliva or whatever, or air. This is through eating already diseased protein or meat with it in it. So prions are very dangerous. All right, our next and kind of like final section is going to be on prokaryotes. And this is that David Goodsell image of what a prokaryotic cell looks like. It's really in depth, but I want you to understand there's a lot more going on in these prokaryotic cells than you think. A lot of people think that bacteria and prokaryotes are really kind of simple, but they're really complex. One more thing is bacteria is not a bad word. The media portrays bacteria like it's something bad, and there are harmful types of bacteria, but I mean, we are made of more bacteria cells than your eukaryotic cells, and there's actually a lot of beneficial bacteria out there as well. But really, the focus in the media gets the bad bacteria, the flesh-eating bacteria, the bacteria that will kill you. But that's only a small portion of all of bacteria. So bacteria is not a bad word. It's actually necessary for life on Earth. Couple just parts of the bacteria cell that I really don't go into a lot of detail, but I like to cover here. The capsule, this is a gel-like coating around the bacteria cell. Fimbriae are just like little projections that will increase the surface area of the cell. You can see here the nucleoid. This is the region where the bacterial chromosome is going to be located. We also going to talk about the flagella. These are just rotating filaments that help propel the cell, and you can see what they look like, but they just basically help move the cell along. One more thing about the kind of structure here is the bacterial cell will have will have plasmids as well as a bacterial chromosome. Both are in the form of circles. We already talked about plasmids before, but you can see what they look like here. Talking about conjugation, this is when bacteria cells will actually share plasmids by kind of just like connecting with each other and, giving the other bacteria cell a version of the plasmid. I'm going to go back a slide. This is called conjugation. This is how they can kind of transfer kind of just genetic information between bacteria cells. It would be like if you and I gave each other a high five and I passed like my genes for eye color to you. Now, that doesn't work in humans, but in bacteria cells, since they're so small, they can actually do this. Now, there are three different types of kind of shapes for bacteria cell. We have bacilla, which is the straight rod. I would say this looks like a pill. The cocci, which is the spherical or circular shape. The spirilla, this is the curved. You can see what that looks like there. And these are just the three main types. And again, there are variations of each one of these, but these are just the three main types. Bacteria are going to grow and divide by something called binary fission. This is basically a type of cell growth. It's asexual reproduction, but it's called the doubling effect, where 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, and so on and so forth. And this is pretty much what we call an exponential growth of the bacterial cells. Now, it will kind of level off because of like limiting factors like space and resources. But this is basically what we're looking at. I'm going to play this quick video because it's really quick. Every 20 minutes, this bacteria is going to divide. So, we go from 2 to 4, from 4 to 8, from 8, it's going to be to 16, from 16 to 32. And within a couple hours, a single bacteria cell can divide many times and create millions to billions of cells. So you can see, what it really becomes pronounced even just after a couple hours or a couple times of division. This is a pretty cool video. So just one cell started here, and now we have many, many, many cells. All right, so we have a couple different types of bacteria. We have obligate anaerobes. These are bacteria that require oxygen to grow. A lot of bacteria are obligate anaerobes, or aerobes. That means they require oxygen. But there are actually some that they're called obligate anaerobes, unable to grow in the presence of oxygen. So what that means is they will die in the presence of oxygen. I know that kind of sounds like crazy to you, but yes, there are bacteria that actually will die in the presence of oxygen. Facultative anaerobes, that means they're able to grow with it without oxygen or with, but they grow much quicker with oxygen. And then aerotolerant anaerobes, these are just bacteria that just grow anywhere. They don't care where they grow, whether with oxygen, without oxygen, it doesn't matter. All right, a couple more things about prokaryotes is they sometimes produce pathogens, and these pathogens cause a toxin or some type of molecule that is going to attack other cells or try to kill other cells or infect other cells. And not every bacteria cell is like this, but these pathogens create something that is kind of toxic to other cells. We have these things that are called antibiotics. They are substances that either kill bacteria by inhibiting their cell wall synthesis or some protein biosynthesis within the bacteria. These were discovered years and years ago, but now we talked about evolution and we talked about how the bacteria cells are actually evolving a ways around these antibiotics. And this is called antibiotic resistance. And this is something that's very scary. This is like climate change is like number one. Antibiotic resistance is like number two for being scary. But these are just ways that your bacteria might evolve around our antibiotics. And, in the future, they're kind of being scared. Science is scared that, our antibiotics won't be able to be used against bacterial infections. And this could actually cause a lot of harm to people. I love the memes here. Yeah. Whenever the bacteria cells develop that antibiotic resistance, yeah, it's, it's a bad thing. A couple more videos. Oh, gram tinning. I don't know if we're going to get to gram tinning this year with a lab, but I always like to try to get to it. This is a stain that's used to identify types of bacteria. Basically, they use just different dyes to identify whether it's gram positive or gram negative. Gram positive bacteria is the thicker cell wall, single membrane, and there is peptidoglycan on the outside. this turns purple. So gram positive is purple bacteria. Gram negative is usually thinner bacteria. They have two membranes. The peptidoglycan is between the membranes, and the dyes turn this bacteria pink. So this is just kind of ways that we can identify cells. I think I show, yep, right here. You can see the gram negative. They have the peptidoglycan in the middle of the two membranes, and the gram positive, they have the peptidoglycan on the outside of the single membrane. All right, last couple slides here. Germ theory of disease. This is where disease are caused by microorganisms. Back years ago, they thought, humans that were bad got diseases, and that's why you were sick. Obviously, that's not the case today. We kind of look at that like, how do people believe that? But it's what people believed. Germs or pathogens cause diseases, and this theory of disease has really revolutionized medicine and helps us understand where diseases come from. And that is the end of chapter 20.